So I want you to imagine something for a second. Picture a physical structure inside your body right now.
Okay.
It is completely hidden from view, obviously, tucked away in the dark. But if you were to somehow extract it, unfold it, and lay it out completely flat, it would actually be larger than a tennis court.
Yeah, the scale is always shocking to people.
Right. And it's not just some massive, passive, empty surface. This structure contains its own entirely independent nervous system.
Which is wild.
It really is. We are talking about hundreds of millions of neurons that are operating completely on their own, constantly reading data. And on top of all that, every single day, this massive hidden tennis court makes literal life or death decisions about your immune system.
It is genuinely one of the most sophisticated, high-stakes, and frankly hostile environments in human biology.
And yet we usually just think of it as a plumbing pipe.
Right, like a tube.
Exactly, like a biological garden hose. You put food in one end, you wait a while, and the rest is history.
Yeah, that's the common perception.
But we are looking at a massive stack of medical sources today for this deep dive. We've got clinical nutrition lectures, advanced gastrointestinal anatomy, physiology papers, detailed pathology exams. And our mission today is to completely tear up that plumbing pipe analogy.
Oh, absolutely. It's a terrible analogy.
Because looking through these sources, the best way I can think to describe your gastrointestinal tract is a highly automated, self-contained chemical processing plant that also doubles as a high-security border checkpoint.
I like that. That is a much more accurate framework. Because it is doing so much more than just, moving things along a conveyor belt. It's analyzing, it's dismantling, sorting, and defending all at the exact same time.
So let's start with the sheer physical architecture of this processing plant. I mentioned a tennis court earlier in the hook. Let's make that real for the listener. Because when I look down at my abdomen, there is physically no space for a tennis court.
Right.
How is that geometric feat actually possible?
Well, it all comes down to the brilliance of fractal geometry.
Fractals.
Yeah. When you look at the human small intestine, you are looking at a tube that is roughly, 2 to 3 meters long in a living person and about 3 to 5 centimeters in diameter.
So like a few feet long, couple inches wide.
Exactly. Now, if it were just a smooth, flat pipe on the inside, like a PVC pipe or a garden hose, it wouldn't have nearly enough surface area to absorb the sheer volume of nutrients you need to stay alive. The math just wouldn't work out.
Because it's just a flat surface.
Right. So biology uses a folding system to drastically multiply the surface area without actually increasing the outer dimensions.
Okay, when you say fractal, I picture those mathematical shapes where you zoom in and it's made at the same shape, and you zoom in again and it's the same shape, just folds upon folds upon folds.
That is the exact principle at play here. So the inside lining of the small intestine, specifically the mucosal lining of the upper sections, which are the duodenum and the jejunum, is not flat.
Right.
It is folded into these large, visible, circular ridges called the folds of Kirkring. You can actually see them with the naked eye.
Folds of Kirkring. Sounds like an alien landscape.
It kind of looks like one. And just by pleating the tissue into these accordion-like folds, the surface area increases by a factor of 3 compared to a flat tube.
Okay, so an accordion fold, that's a 3 times multiplier right off the bat. But 3 times the surface area of a 3-meter tube is still not anywhere close to a tennis court.
Far from it. So covering every single square inch of those folds of Kirkring are millions of tiny finger-like projections called villi.
Like little hairs?
Kind of. If you looked at it under a magnifying glass, the lining would look kind of like the surface of a shag carpet.
Okay, gross, but I get the visual.
Yeah, and each individual villus projects about 1 millimeter out from the surface of the mucosa. And these aren't just, dead hairs. They are highly vascularized living structures.
Meaning they have blood supply.
Exactly. They are packed with capillary beds and tiny lymphatic vessels. Covering the folds with these villi increases the total surface area by another factor of 10.
Oh, wow. Okay, so the 3x from the folds is multiplied by the 10x from the villi. We are at a 30-fold increase over a flat tube. Still not a tennis court, though.
Right. Here's where we cross into the microscopic magic.
I'm ready.
The border of every single one of those villi is lined with individual epithelial cells. And the top of each of those cells, the side facing the inside of the tube, is covered in even smaller projections called microvilli.
So hairs on the fold.
Yes. This is what physiologists call the brush border. There are roughly 600 microvilli on a single cell.
600.
Yeah, and each one is only about 1 micrometer long. This microscopic brush border increases the surface area a further 20 folds.
It's just mind-blowing when you stack it all up.
Yeah.
Folds on top of fingers on top of microscopic hairs. So let's run that final math for the listener.
Go for it.
A 3-fold increase for the folds of Kirkring, times a 10-fold increase for the villi, times a 20-fold increase for the microvilli brush border. That is a 600-fold increase in total absorptive surface area compared to just a simple, flat anatomical tube.
Yep, the math scales incredibly fast. And when you calculate the total surface area of this highly specialized microscopic construction, it comes out to roughly 250 square meters.
Which is insane.
It is. To put that in perspective, a standard singles tennis court is about 195 square meters.
Yeah.
So your small intestine literally has a larger surface area than a tennis court, all coiled, folded, and packed neatly inside your abdominal cavity.
That physically hurts my brain to think about, but it's amazing. Let me play devil's advocate for a second, though.
Sure.
If you're the listener, you might be wondering, practically speaking, why go through all this trouble? Like, why do we need an entire tennis court of surface area inside of us just to eat a sandwich?
Well, because absorption is fundamentally a game of physical contact. Nutrients don't just magically teleport from the center of the tube into your bloodstream.
Right, they have to touch the walls.
Exactly. About 95% of all absorption takes place in the very first 2 segments of the small intestine. That's the duodenum, which is short, about 30 centimeters long, and the jejunum, which is the next 1 to 2 meters.
Okay.
And to get all the carbohydrates, amino acids, and fats out of your food, that food, which by the time it reaches the intestine is a thick, semi-fluid paste called chyme, has to physically touch the walls of the intestine. The larger the surface area, the more molecular contact you get.
Which brings up a mechanical problem. I mean, if we just drop food down a pipe, gravity would pull it straight through. We're talking about motility now, right?
Yeah.
The gut can't just rely on gravity to ensure this thick paste smears evenly over 250 square meters.
No, not at all.
It has to actively push and mix the food.
Right. And the gut uses 2 main types of movement to solve that exact problem, driven by 2 distinct layers of smooth muscle.
What are the layers?
There's a circular layer that wraps around the tube and a longitudinal layer that runs lengthwise. So first you have peristalsis. This is the propulsive movement.
The one that pushes it forward.
Yeah, I always picture squeezing a tube of toothpaste from the bottom up. The circular muscles contract right behind the bolus of food, pushing it forward at a rate of about 0.5 to 2.0 centimeters per second.
Okay.
It's fastest in the upper intestine, and then it naturally slows down as it gets further along.
But wait, if the gut just did peristalsis, like if it just shot the food straight through, like squeezing a toothpaste tube, it wouldn't have time to absorb anything. It would just slide right past the tennis court.
Exactly. Which is why the second type of movement is arguably the most critical for survival, and that's segmentation.
Segmentation.
These are mixing contractions. Instead of a sweeping, progressive wave pushing things forward, the circular muscles contract intermittently in different isolated spots.
So not a wave.
No. It makes the intestine temporarily look like a string of linked sausages. These contractions fire off about 8 to 12 times a minute.
So it's not pushing it forward, it's just squishing it in place.
It squishes it back and forth. The chyme is forced backward just as much as it is forced forward. And this constant rhythmic churning accomplishes 2 really vital things.
What's the first?
First, it mixes the chyme thoroughly with all the digestive juices and enzymes so the chemical breakdown can actually happen.
Makes sense.
But secondly, and most importantly for our tennis court analogy, it constantly cycles new chyme from the center of the tube out to the edges.
Oh, I see.
It prevents a stagnant layer from forming along the wall, ensuring every single molecule eventually gets pushed against those microscopic brush border microvilli.
It's forcing turbulent flow instead of laminar flow.
Yeah.
It basically wants chaos in there so everything touches the wall at some point.
Exactly.
And it's a very deliberate, timed process. The sources say food usually spends about 3 to 10 hours just navigating the small intestine. But here is where it gets really interesting to me as a non-scientist.
Oh, yeah.
Yeah, because to coordinate all of this, the peristalsis pushing it forward, the segmentation churning it up, the precise timing of the muscle layers, you need a central computer. You need a brain. But the gut doesn't wait for the brain in your head to tell it what to do, does it?
It can't. The math of the data processing makes it completely impossible.
How so?
If your central nervous system had to consciously receive stretch signals, calculate transit times, coordinate the dual muscle layer contractions, and manage the chemical mixing of every single bite of food across 250 square meters of tissue.
I'm getting overwhelmed just hearing that.
Right. Your brain would be entirely overwhelmed. You literally wouldn't have the processing power to walk, talk, or breathe. So, evolutionary design gave the gut its own autonomous brain.
The second brain.
Yes, it is officially called the enteric nervous system, or the ENS.
I always thought second brain was just, a pop science buzzword, but looking at the data, it's quite literal.
Oh, it absolutely earns the title. The ENS contains somewhere between 400 and 600 million neurons.
It is massive.
It's a massive network capacity, roughly equivalent to the number of neurons in your spinal cord. It allows the gut to operate entirely independently via highly complex local reflexes.
So it doesn't need the brain at all.
Well, it can still communicate bidirectionally with the central nervous system. The vagus nerve is the main highway for that. But if you were to sever the vagus nerve, the gut would continue to digest food. It is functionally autonomous.
The analogy I keep coming back to for the ENS is like a highly advanced smart home network.
Okay, I like where this is going.
If you walk into a room and a motion sensor turns on a light, the sensor doesn't need to ping a cloud server in California to ask permission. It handles the logic locally. The ENS is doing that, but instead of motion sensors, it has complex chemical sensors tasting the food, stretch receptors feeling the volume, and muscle actuators running the mixing programs.
That analogy holds up perfectly. And to run a factory that size, this smart home network is actually divided into two primary subnetworks, or plexuses, and each has a very specific jurisdiction.
Yeah, what are they?
First, you have the myenteric plexus, historically known as Auerbach's plexus. If you look at its physical location, it is sandwiched right between the circular and longitudinal smooth muscle layers.
Between the muscles.
Right. And because of where it sits, its primary function is controlling motility. It is the network that dictates the peristalsis and the segmentation we just broke down.
So Auerbach's plexus is basically the mechanical engineer. It handles the physical movement of the factory floor.
Spot on. And while Auerbach's is managing that mechanical movement, the chemical side is being handled right next door by the submucosal plexus, or Meissner's plexus.
Meissner's, okay.
This one is located deeper into the tissue in the submucosal layer, just under the inner lining. Its primary function is chemical and sensory. It controls the local secretion of digestive juices, it alters local blood flow to match the digestive demand, and it regulates absorption across the epithelial cells.
Okay, so Meissner's plexus is the chemical engineer.
Exactly.
The mechanical and chemical engineers work together entirely locally to run this massive automated plant. But to run the plant, these engineers rely on a team of chemical shift managers. And reading through these sources, the way the gut shifts gears based on whether you're eating or not eating is truly a marvel.
It really is.
Let's dig into the hormonal control room.
This is a fascinating area of physiology that dictates a lot of our daily comfort, actually. The gut basically operates in two distinct, mutually exclusive modes, the fasting state and the fed state. And these states are entirely dictated by specific hormones released by the cells lining your intestine.
Let's start with the fasting state, because I think a lot of people, myself included until I read this, assume that when you aren't eating, your gut is just, like, empty and asleep.
A lot of people think that.
But it's actually doing something incredibly important.
It is running a critical maintenance protocol. During the fasting state, which is the period between meals when there are no nutrients in the lumen, and the pH of the duodenum is relatively alkaline, specialized endocrine cells called M cells wake up.
M cells.
Right. And they release a hormone called motilin into the bloodstream.
And just a quick time out for the listener, because the sources throw a real curveball here. These are M cells in the upper gut, the duodenum and proximal jejunum. We're going to talk about a totally different kind of cell later that is also, unfortunately, called an M cell, so we need to keep that straight.
Yes, very important distinction.
But these upper gut hormonal M cells release motilin. What is motilin's job?
Motilin is the master key that initiates something called the migrating motor complex, or the MMC. In some older literature, you might see it called the migrating myoelectric complex. But you can think of the MMC as the gut's biological housekeeper.
Okay, the housekeeper. I love this. Walk me through the cleaning cycle.
When motilin binds to receptors on the smooth muscle of the gut, it triggers a traveling wave of electrical and muscular contraction. This wave originates way up in the pacemaker region of the stomach, specifically an area called the greater curvature of the antrum, and it sweeps distally or downward all the way through the small intestine.
And this isn't just a quick random sweep, right? It's a highly structured cycle.
It is a very deliberate 90 to 120-minute cycle divided into 4 distinct phases.
What's phase one?
Phase 1 is quiescence, lasting 40 to 60 minutes, just complete motor silence, nothing is happening. Phase 2 brings irregular, intermittent contractions for another 20 to 40 minutes, and that's accompanied by some mild gastric acid and pepsin secretion.
Okay, building up to something.
Exactly. Because phase 3 is the climax. This is the actual housekeeper phase. For about 5 to 10 minutes, you get a rapid, sustained burst of intense, rhythmic contractions at absolute maximum frequency.
How fast are we talking?
We are talking about 3 contractions per minute in the stomach and up to 12 per minute in the duodenum. It is a powerful, sweeping wave. And then finally, phase 4 is just a quick 5-minute decline back into motor silence.
So every hour and a half to 2 hours that you don't eat, you get this intense 10-minute deep clean that pushes downward at about 5 to 10 centimeters per minute.
Yes.
If you're listening to this and your stomach just loudly growled because you skipped lunch,
Yeah.
that's phase 3 of the MMC happening right now.
That is exactly what that sound is.
But why is this sweeping motion so critical? What actually happens if the housekeeper goes on strike?
Without the MMC, the small intestine is in severe trouble. Its primary function is gastrointestinal cleansing. It physically sweeps undigested debris, metabolic waste, and sloughed off dead epithelial cells out of the small intestine and into the large intestine, or colon, for excretion.
Right.
But even more importantly than sweeping debris, it sweeps bacteria.
Ah, right, because as we'll discuss later, the colon is teeming with literally pounds of bacteria. But the upper small intestine is supposed to be a relatively sterile environment.
Precisely. The MMC is the physical barrier that prevents those colon bacteria from creeping upward. When the MMC is impaired, say, due to nerve damage like the autonomic neuropathy we sometimes see in diabetes, which can cause delayed gastric emptying or gastroparesis, those phase 3 sweeping contractions become really weak, or they vanish entirely.
And the bacteria just march upward. This is the mechanism behind SIBO, right?
Yes, small intestinal bacterial overgrowth. When the housekeeper stops sweeping, bacteria from the colon translocate proximally, take root in the nutrient-rich small intestine, and start fermenting your food prematurely.
That sounds awful.
It is. This leads to severe bloating, abdominal pain, and massive malabsorption because the bacteria are eating your nutrients before you can absorb them. Altered MMC cycling is also heavily implicated in IBS or irritable bowel syndrome.
This brings up a really practical question for the listener. The sources note that the physical ingestion of food is the absolute strongest inhibitor of the MMC.
It is.
The moment vagus nerve stimulation happens, or the stomach stretches, or digestive hormones are released, the body instantly aborts the MMC and switches to the fed state.
So wait, if you're listening to this while snacking on a handful of almonds, did you just hit the pause button on your gut's cleanup crew? Like, does the modern habit of grazing all day prevent our gut from cleaning itself?
It absolutely does. If you are constantly eating every hour or two, you never reach that crucial fasting window required to trigger Modlin and launch phase 3 of the MMC.
Your gut is perpetually stuck in the fed state. It's mixing, it's churning, it's absorbing, but it is never sweeping. Biologically, the human system requires a 2 to 3 hour fasting window between meals just to reset and clean the factory floor.
That is such a massive aha moment for me.
Yeah.
We hear about fasting all the time as this extreme weight loss or longevity trend, but biologically, just spacing out your normal meals and not snacking is a baseline requirement for basic gut hygiene.
It really is fundamental physiology.
It's so crucial that clinically, if someone's MMC is paralyzed, doctors will sometimes prescribe a specific antibiotic, a macrolide called erythromycin. But they aren't using it to kill bacteria, are they?
No, the antibacterial properties are completely secondary in this context. They're using erythromycin off-label as a prokinetic agent.
Prokinetic meaning it makes things move.
Exactly. Erythromycin happens to be a potent Modlin receptor agonist. Structurally, it mimics Modlin enough that it binds to the receptors in the gut and artificially forces the smooth muscle to trigger those phase 3 sweeping contractions.
That is amazing.
Right. In patients with severe postoperative ileus, where the gut just freezes after surgery or gastroparesis, it acts like a chemical jumper cable to restart the housekeeper.
That is just wild. Okay, so that's the fasting team. The housekeeper's squeaking up. Now, let's say you do finally sit down and eat a meal. The food arrives. Motilin is immediately suppressed. The housekeeper puts the broom away. Who takes over the control room now?
The moment food enters the duodenum, a highly coordinated fed team of hormones takes charge.
And what's their job?
Their job is entirely different. They aren't cleaning. They need to aggressively manage digestion, protect the delicate intestinal lining from stomach acid, and prepare the body's entire metabolic network for an influx of nutrients.
So who are the players?
The big three chemical managers here are CCK, secretin, and GIP.
Let's break these down because the coordination is fascinating. I want to use a real-world example here. Let's say you just ate a big, greasy cheeseburger.
A classic example.
It spends some time in your stomach, and then this highly acidic, fat-heavy paste gets squirted into your duodenum. It's basically chemical chaos. But the gut responds with precision. Let's start with the fat and the protein from the burger patty and the cheese. Who handles that alert?
That alert is handled by cholecystokinin, or CCK.
CCK.
It is synthesized and released by specialized enteroendocrine cells called I cells, which are located in the mucosa of the duodenum and proximal jejunum.
And what triggers them?
The moment these I cells detect the physical presence of fats, specifically triglycerides and long-chain fatty acids, and partially digested proteins from the meat, they flood the local bloodstream with CCK.
And CCK is like the ultimate multitasker. The notes literally call it the gallbladder and satiety hormone, but it seems to do even more than that.
It acts as the grand conductor for digestion. Its actions are incredibly coordinated across multiple organ systems. First, it targets the gallbladder.
Okay, what does it tell the gallbladder?
CCK provides a powerful stimulus for the gallbladder to contract and squeeze its stored, concentrated bile into the common bile duct. Fats don't dissolve in water, so they need those bile salts to emulsify them.
Got it. Bile for the fats. What else?
Second, CCK travels to the pancreas and stimulates the pancreatic acinar cells to release a massive payload of digestive enzymes. We're talking lipase for the fats, amylase for the carbs, and proteases for the proteins.
So it sees the fat and immediately calls in the bile to dissolve it and the enzymes to chop it up. But those chemicals are stuck in ducts, right? How do they get into the actual gut tube?
That is CCK's third action. It actively relaxes the sphincter of Oddi.
Sphincter of Oddi.
Yeah, this is the crucial muscular valve located at the ampulla of Vater, which is the exact point where the bile duct and pancreatic duct enter the duodenum.
Oh, I see.
By relaxing the sphincter, CCK opens the floodgates, allowing the bile and enzymes to physically enter the gut and mix with the cheeseburger chyme.
It's like a highly efficient traffic cop. It sees a massive convoy of fat approaching, calls for the chemical support, and opens the intersection so they can actually meet. But it also has to manage the flow of traffic coming from the stomach, right?
Yes, and this is a vital protective mechanism. CCK heavily slows down gastric emptying.
How does it do that?
It sends signals that partially contract the pyloric sphincter, the valve between the stomach and the duodenum. Essentially, the small intestine is sensing the fat and telling the stomach, whoa, there is a massive amount of fat here, and fat takes a very long time to emulsify and digest. Slow down.
Oh, it prevents a bottleneck.
Exactly. It prevents the duodenum from being overwhelmed beyond its capacity. And as a final overarching effect, CCK acts on the vagus nerve and directly on the hypothalamus in your brain to signal satiety. It tells your conscious mind that you are full and should probably stop eating.
Okay, so I totally get how CCK handles the fat and protein from our cheeseburger, but I'm stuck on a glaring environmental hazard here.
What's that?
That meat spent the last couple of hours in a bath of intense stomach acid. So this chyme entering the small intestine is highly, highly acidic. If the small intestine is so delicate with its tennis court villi, wouldn't this acidic paste literally burn a hole right through the wall? How do we survive every meal?
We survive because of the second manager. Secretin. Okay. Secretin is released by a different set of sensors called S cells, which are densely packed in the crypts of the duodenum.
Okay, S cells for secretin, I cells for CCK. Easy enough to remember.
Right. And the primary trigger for the S cells is entirely different from the I cells. They don't care about fat at all. They care about pH.
So they are acid sensors.
Exactly. They are triggered by a high proton load hydrochloric acid. When the S cells detect this severe acid drop, they dump secretin into the blood, which basically acts as the body's ultimate acid neutralizer.
What's its actual mechanism, though? Does it just go back to the stomach and turn off the acid pump?
It does mildly inhibit gastric acid production as a negative feedback loop, but its primary and most critical target is actually the pancreas, specifically the pancreatic ductal cells.
What does it do to them?
Secretin binds to these cells and forces them to secrete a massive volume of watery fluid that is incredibly rich in bicarbonate.
Bicarbonate, literally the active ingredient in baking soda.
Precisely the same chemical. This bicarbonate-rich fluid floods down the duct and into the duodenum, where it mixes with the stomach acid. It rapidly raises the pH of the incoming chyme from a highly dangerous acidic pH of around 2 up to a much safer, slightly alkaline pH of 7 or 8.
So it's literally a biological fire extinguisher.
Yes, and it serves two purposes. First, as you noted, it physically protects the duodenal mucosa from acid burn and ulceration.
Which is critical.
But second, it is chemically necessary for digestion to even continue. The powerful pancreatic digestive enzymes that CCK just called in, they can only function in a neutral or slightly alkaline environment.
Oh.
If the acid isn't neutralized by secretin, those enzymes are permanently deactivated and you digest nothing. Secretin also gets the liver in on the action, stimulating it to produce more of the watery, bicarbonate-rich component of bile to help with the neutralization.
This level of coordination is just stunning. So the I cells sense fat and call in CCK to get bile and enzymes. The S cells sense acid and call in secretin to get baking soda from the pancreas. We've handled the fat, the protein, and the acid. But looking at this cheeseburger, we haven't touched the bun. How does the body handle the massive carbohydrate load?
That is managed by the third member of the FED team, GIP, or glucose-dependent insulinotropic peptide.
GIP.
It's released by K cells located throughout the duodenum and jejunum, and they fire in direct response to the presence of sugars and, to a lesser extent, some fats.
Now, in some older biology textbooks, I've seen GIP referred to as gastric inhibitory peptide. Is that the exact same thing?
It is the exact same hormone, yes. Early on, scientists realized it mildly inhibited gastric acid secretion and slowed gastric emptying, similar to CCK, hence the old name.
But modern physiology heavily favors the new name because its primary metabolic role is far more important. GIP is what we call an incretin.
I've heard the word incretin thrown around a lot lately with all these new weight loss drugs. What does an incretin actually do in a normal, healthy system?
An incretin hormone acts as an early warning system for the entire metabolic network. When GIP detects glucose in the gut tube, it travels through the bloodstream straight to the pancreas.
Back to the pancreas again.
Yes, but it bypasses the digestive enzyme cells and goes straight to the endocrine beta cells. It tells those beta cells to start producing and releasing insulin before your blood glucose levels even begin to rise systemically.
That is unbelievably smart. It's prepping the body for the sugar rush. It's sensing the bun in the factory and saying, hey, a massive payload of glucose is currently being processed. It's going to hit the bloodstream in about 10 minutes, get the insulin ready and waiting at the cellular door so we don't suffer a dangerous spike in blood sugar. That's exactly it. And that anticipatory insulin release allows for rapid, safe glucose uptake by your peripheral cells the precise moment the sugar crosses into the blood. So when you zoom out and look at the FED, Team CCK, secretin, and GIP, what is truly beautiful here is the simultaneity.
Yeah, they all work at once.
When that cheeseburger hits the duodenum, I cells, S cells, and K cells are all firing at once, perfectly managing fat, acid, and sugar, while completely shutting down Matilin and the housekeeper.
It's an incredible chemical symphony.
Yeah.
But these hormones are just the shift managers giving orders, right? They don't actually break down the food themselves.
Correct.
Let's move on to the factory floor and look at the actual demolition crew. The secretion, digestion, and absorption processes. To understand this, we actually have to take a step back from the small intestine and look at the stomach. The stomach is famous for one thing, acid. Just how strong is this stuff?
It is remarkably dangerously potent. The parietal cells embedded in the stomach wall secrete an isotonic hydrochloric acid solution at a concentration of 160 millimolar.
What does that mean in terms of pH?
To put that in perspective, this equates to a pH of about 0.8 at the exact point of secretion. By the time it mixes with the food and mucus in the stomach cavity, the overall gastric pH hovers around 2.0.
A pH of 0.8 is essentially battery acid. If you dropped a steel nail into a vat of that, it would dissolve. Why on earth do we need a reservoir of something so corrosive inside our bodies?
For several evolutionary and biochemical reasons. First, it is a primary immune barrier. It instantly kills the vast majority of pathogenic organisms, bacteria, and viruses that hitch a ride on the food we eat.
Okay, sterilization.
Second, it chemically denatures food proteins.
Denatures.
It physically uncoils the complex three-dimensional folded structures of proteins, exposing their molecular bonds so enzymes can actually reach them. Third, that high acidity is required to properly absorb vital minerals like iron and calcium. And finally, and perhaps most importantly, it serves as the environmental activator for a critical protein-destroying enzyme.
Right, because I was thinking, if the stomach just produced a highly corrosive, protein-destroying enzyme natively alongside the acid,
Okay.
it would just digest its own walls.
And it absolutely would. This brings us to a brilliant biological failsafe, the concept of zymogens, which are inactive proenzymes.
Inactive proenzymes, how do they work?
The chief cells of the stomach wall secrete an inactive precursor molecule called pepsinogen. While it is inside the cell, it is completely harmless. It cannot digest anything.
Okay.
But the moment pepsinogen gets secreted and comes into contact with that extreme hydrochloric acid in the stomach lumen, the acid cleaves off a piece of the molecule, instantly converting it into the active, aggressive protease called pepsin.
I love this concept. I picture it like a munitions factory that manufactures highly volatile, highly corrosive explosives, but they ship them out onto the floor without the detonators attached.
Exactly.
The stomach cells ship inactive pepsinogen, and the pool of acid is the environmental detonator that arms it only once it's safely in the middle of the stomach, far away from the delicate cellular machinery.
That's a highly accurate way to visualize it. And once activated, pepsin is a crucial enzyme because it specifically degrades the tough, collagenous connective tissue fibers found in meat.
Oh, nice.
Once pepsin dismantles those tough fibers, other proteases can effectively access and digest the remaining animal protein. Usually, the stomach enzymes and acid manage to hydrolyze or break down about 10 to 20% of the ingested proteins before the chyme moves on.
Which brings us to the logistics of gastric emptying. The stomach is churning this acidic, pepsin-filled soup. How does it know when it's time to open the pyloric sphincter and let this slurry into the delicate small intestine?
Gastric emptying is a tightly regulated, negotiated process between the stomach and the small intestine. It usually takes anywhere from 1 to 4 hours for a normal meal. Yeah, and it's initially driven by the stomach stretching and hormonal signals like gastrin, which promote emptying, but the strongest regulators are actually negative feedback loops coming from below, from the duodenum. The duodenum has a vast array of receptors that constantly monitor the quality and volume of the incoming chyme.
So the duodenum is basically running quality control. What specific metrics is it looking for before it allows more food in?
It monitors acidity, distension, and osmolarity. We already mentioned that if the chyme is too acidic, secretin fires and simultaneously slows the stomach down.
Right, the fire extinguisher.
If the physical volume stretching the duodenum is too high, it triggers the enterogastric reflex via the enteric nervous system, tightening the pyloric sphincter to prevent dumping too much volume at once.
The sources also list several external factors that affect how fast the stomach empties. Fluid volume is a big one, right? Like, if you drink a massive glass of water on an empty stomach, it empties exponentially faster than just a few sips.
Yeah.
But what about the composition of the meal itself?
Energy density is a major dictating factor. Fat is a remarkably strong inhibitor of gastric emptying.
Which makes sense based on the CCK stuff we talked about.
Exactly. It makes evolutionary sense because fat takes a very long time to emulsify and digest. The intestine needs time to process it. But even the concentration of carbohydrates matters deeply. A standard sports drink with a 2% carbohydrate solution will empty slightly slower than plain water, but a highly concentrated solution of 8% or more significantly inhibits emptying.
The sources also mention osmolarity playing a huge role. Like, if a drink has a very high osmolarity, meaning a very high concentration of solute particles, like greater than 500 millimoles per liter, it will drastically reduce gastric emptying.
Yes.
And worse, if it does get into the intestine, it will actually draw water out of your body's bloodstream and into the gut to dilute it. We'll come back to why that's a total nightmare for endurance athletes later.
Oh, it really is.
There are also some fascinating lifestyle mentions in the sources. Psychological stress. When you are stressed, sympathetic hormones like epinephrine reduce blood flow to the GI tract, which effectively slows gastric emptying to a crawl. And apparently women generally have slightly slower gastric emptying rates than men, though it fluctuates with the hormones of the ovulation cycle.
Right. And the overarching principle governing all of these variables is simply protection and efficiency. If the stomach delivers chyme too rapidly, the small intestine won't have sufficient time or enzymes for complete digestion and absorption, and vital nutrients will be lost.
Okay, so the stomach negotiates with the duodenum and eventually empties. We are back in the small intestine. The CCK and secretin alarms have been pulled. Now the real heavy hitters arrive from the accessory organs. Let's talk about the exocrine pancreas. What is it actually doing?
The exocrine pancreas is a large lobulated gland situated just beneath and behind the stomach. It is an absolute powerhouse of chemical synthesis.
How much is it making?
It secretes 1 to 2 liters of fluid every single day. We already talked about the secretin-driven bicarbonate that acts as the fire extinguisher, but the CCK-driven enzyme secretion is where the true chemical demolition happens.
And reading the notes, it uses that same zymogen explosive without a detonator trick that the stomach uses, but on an even grander scale, right?
On a much grander and much more dangerous scale. The pancreatic juice is incredibly rich in a whole family of enzyme precursors. Trypsinogen, chymotrypsinogen, proelastase, procarboxypeptidase.
That's a mouthful.
It is. But these are all completely inactive. If they were to activate while still inside the pancreas, the organ would literally digest itself from the inside out.
Yikes.
Yeah, a catastrophic, life-threatening condition known as acute pancreatitis. The body is so paranoid about this that the pancreas even secretes a specific protease inhibitor alongside the zymogens, just in case a tiny bit of trypsinogen accidentally activates prematurely. It's a chemical safety catch.
So we have this massive payload of inactive explosives flowing down the pancreatic duct. How do they finally get activated once they safely reach the intestine? What is the detonator here?
It relies on a beautifully localized physical mechanism. Remember that brush border, the microvilli lining the tennis court?
Yeah.
Embedded directly into the membrane of those microvilli is a specific enzyme called enteropeptidase or enterokinase. It is physically anchored to the wall.
Right.
When the inactive pancreatic trypsinogen flows out of the duct and washes over the brush border, it physically touches the enteropeptidase. That contact cleaves a peptide bond, converting the inactive trypsinogen into the highly active enzyme, trypsin.
And trypsin is the master detonator.
Exactly. Or, to avoid the repetitive phrase, you could view trypsin as the domino that knocks down the rest.
Oh, I like that.
Once you have active trypsin in the lumen, it immediately goes to work activating all the other proenzymes. It turns chymotrypsinogen into chymotrypsin, procarboxypeptidase into carboxypeptidase. It creates a massive rapid cascade of active proteases that swiftly chop the long, complex polypeptide chains of your cheeseburger down into tiny tripeptides, dipeptides, and single amino acids, which are finally small enough to be absorbed.
That cascade is brilliant. And the pancreas isn't just handling protein. It also drops in pancreatic amylase for the carbs. Now, I know we have salivary amylase in our mouths, which starts breaking down the bun while we chew. But by the time that bun hits the stomach acid, that salivary amylase is mostly deactivated, right?
Yes, salivary amylase cannot survive the 0.8 pH of the stomach. So the pancreatic amylase picks up the slack in the neutral environment of the small intestine, rapidly hydrolyzing complex starches into smaller sugars like dextrins and maltose.
And then what?
And then finally, embedded right next to the enteropeptidase on the brush border, are specific sugar enzymes, lactase, sucrase, and maltase, that break those smaller sugars down into their final absorbable monosaccharide forms: glucose, galactose, and fructose.
All right, proteins and carbs are handled, but we are missing the hardest macronutrient. Pancreatic lipase handles the fats. But reading the sources, pancreatic lipase has a massive physical problem. Fats are hydrophobic, they clump together in water. But lipase is water-soluble. It can only work on the very outer surface of a massive fat globule. This is where the liver and gallbladder become absolutely essential to survival, don't they?
They do. This is where we return to bile. Bile is synthesized by the liver and consists of water, electrolytes, cholesterol, lecithin, bilirubin, and critically for digestion, bile salts.
And the gallbladder stores it.
Right. The gallbladder acts as a reservoir, storing and dehydrating this bile, concentrating it up to 5 to 10-fold. When CCK hits the gallbladder, it contracts, and this highly concentrated bile shoots into the duodenum.
And what do these bile salts actually do to the clumped up fat from the cheeseburger?
They act exactly like biological dish soap. If you have a greasy pan and drop water in it, the grease clumps together. Drop dish soap in, and the grease instantly shatters into tiny droplets.
So it shatters the fat.
Yes. The bile salts arrange themselves with phospholipids and the dietary fatty acids into microscopic structures called micelles. Micelles are well-defined, disc-like shapes. The fat-soluble part of the bile salt faces inward, binding to the fat core, and the water-soluble part faces outward. This allows the entire micelle structure to exist smoothly and remain suspended in the watery environment of the gut.
It emulsifies the fat into microscopic droplets, and by shattering one massive fat globule into a million tiny micelles, you vastly increase the surface area for the pancreatic lipase to actually reach the triglycerides and break them down.
Exactly.
And I was reading that pancreatic lipase actually requires a tiny helper molecule, a cofactor called colipase, just to anchor it to the slippery lipid-water interface so it can do its job.
Right. And without the bile salts forming these micelles and the colipase anchoring the enzyme, fat absorption is severely compromised. In a healthy system with adequate bile, fat absorption is highly efficient, almost 97% complete.
And without bile.
Without bile, only about 50% of fatty acids are absorbed. The rest travels down to the colon, leading to severe nutritional deficiencies and a miserable condition called steatorrhea, which we will explore when we get to pathology.
But wait, if bile is this critical, the body doesn't just use these bile salts once to digest one meal and then flush them down the toilet? Right. That seems incredibly wasteful.
It would be energetically impossible. The body synthesizes bile salts from cholesterol, which is a resource-intensive process. It cannot afford to synthesize a completely new batch of bile salts for every single meal.
So it recycles them.
Yes, evolution provided an incredibly efficient recycling mechanism called the enterohepatic circulation.
Breaking down the word. Entero meaning intestine, hepatic meaning liver.
You got it. The micelles ferry the digested fats to the brush border, where the fatty acids simply diffuse into the intestinal cells. The bile salts themselves, however, do not enter the cell there.
Where do they go?
They travel all the way down the entire length of the small intestine to the very last segment, the terminal ileum. There, highly specialized active transporters grab the bile salts and pull them into the portal blood, which carries them straight back to the liver.
Wow.
The liver extracts them from the blood and immediately resecretes them into the bile ducts. Through this continuous loop, roughly 94% of bile salts are continuously recycled.
It's an incredibly tight, closed-loop system. Okay, so we finally demolished the cheeseburger into its smallest building blocks.
Yeah.
Single amino acids, monosaccharides like glucose, and free fatty acids. They're just sitting on the tennis court. How do they actually get across the wall and into the body? It turns out they take very different routes.
They do. Let's start with carbohydrates and proteins. The absorption of glucose and galactose is not a passive process. It requires energy and active transport. They use a specific carrier protein embedded in the cell membrane called SGLT1, the sodium-dependent glucose transporter.
And as the name implies, it requires sodium to function.
Yes, and the math here is specific. For every single molecule of glucose that is brought into the cell, 2 sodium ions must be transported into the epithelial cell along with it. This is called secondary active transport.
Why does sodium want to go in so badly?
The only reason sodium wants to enter the cell so badly is because a sodium-potassium pump on the far side of the cell is constantly burning cellular energy ATP to pump sodium out, creating a massive concentration gradient. The glucose essentially hitchhikes on the energy of that sodium gradient.
Okay, that makes sense.
Fructose, interestingly, doesn't use this system. It uses a different sodium-independent transporter called GLUT5. Amino acids also largely rely on a similar sodium-dependent active transport mechanism.
Okay, so the cell burns energy to pump the carbs and proteins across the border. Once they cross the cell, where do they go?
They exit the basolateral side of the cell, the side facing the body, and diffuse into the local capillary beds inside the villi. These thousands of tiny capillaries converge into larger and larger veins, eventually forming the hepatic portal vein.
The portal vein.
You can think of the portal vein as a direct, high-security express highway that shuttles these raw, newly absorbed nutrients straight to the liver for immediate chemical inspection, processing, storage, or distribution to the rest of the body.
So carbs and proteins take the blood highway straight to the liver. But reading further, fats take a completely different, much weirder path, which I find fascinating.
The routing for fats is entirely distinct and much more complex. When the long-chain fatty acids diffuse into the epithelial cell from the micelles, they aren't sent to the blood.
What happens to them?
Instead, the cell's internal machinery takes those free fatty acids and painstakingly reassembles them back into complete triglycerides in the endoplasmic reticulum. It then packages these reassembled triglycerides, along with cholesterol and phospholipids, into massive protein-coated transport spheres called chylomicrons.
And these chylomicrons are physical behemoths, right? They're too big to fit through the tiny pores of standard blood capillaries.
Exactly. So instead of blood, they enter the lymphatic system. If you recall our anatomy from earlier, inside the center of every single microscopic villus is a blind-ended lymphatic vessel called a lacteal. The massive chylomicron chylomicrons are absorbed into these lacteals. They travel slowly through the lymphatic vessels, entirely bypassing the liver initially, and are eventually dumped directly into the systemic blood circulation near the heart, specifically at the left subclavian vein.
That is just wild to me. Carbs go to the liver for strict inspection, but fats take the lymphatic back roads, bypass the liver's security checkpoint, and get dumped straight into the heart and the rest of the body. Now, the sources do mention an exception, though. Short and medium-chain fatty acids are small enough and water-soluble enough that they just diffuse right into the portal blood with the carbs.
That's a crucial clinical distinction. It is often highly relevant in medical nutrition therapy for patients who have damaged lymphatic systems or lack bile, because you can feed them medium-chain triglycerides, MCT oil, and they can absorb them directly into the blood without needing micelles or chylomicrons.
Before we leave absorption, we need to talk about the sheer volume of water the gut handles. It's staggering.
The fluid dynamics are truly immense. About 99% of water absorption takes place in the small intestine, primarily the duodenum and jejunum, purely by simple diffusion driven by osmosis.
How much water are we talking?
To give you context, the gut sees a massive amount of water daily. You might only drink 2 liters, but your salivary glands, your stomach, your pancreas, and your liver add up to roughly 7 liters of endogenous secretions every single day.
7 liters inside the body.
Yes. So the small intestine is actively absorbing up to 9 liters of water daily. It simply follows the solutes. As all that sodium and glucose we just talked about are actively pumped into the body, water gets physically pulled along with them through the osmotic gradient.
And this explains a major pathology. If you have a massive influx of a highly concentrated, high osmolarity substance sitting in the gut like undigested lactose or a hyperconcentrated energy drink, it reverses the osmotic gradient. It pulls water out of the body's bloodstream and into the gut lumen, causing sudden, explosive osmotic diarrhea.
Which is incredibly dangerous. When that happens, you aren't just losing the water you drank, you are losing those 7 liters of vital internal secretions. It can lead to life-threatening dehydration and electrolyte collapse very quickly.
And as for vitamins, fat-soluble vitamins A, D, E, and K are absorbed just like the fats, right? They get incorporated into the micelles and then packaged into the chylomicrons. So if you have a bile deficiency, you aren't just missing out on fat calories. You are developing severe, systemic vitamin deficiencies. Vitamin K, for instance, is vital for blood clotting.
Meanwhile, most water-soluble vitamins diffuse easily in the upper small intestine. However, vitamin B12 is a glaring, complex exception.
Oh yeah, B12 is weird.
Its absorption is highly convoluted. It requires a specific binding protein called intrinsic factor. And intrinsic factor is secreted all the way back in the stomach by the exact same parietal cells that make the hydrochloric acid.
But it doesn't get absorbed there.
No. The actual absorption of the B12 intrinsic factor complex doesn't happen in the stomach, or the duodenum, or the jejunum. It only happens at the very end of the line via highly specific receptors in the terminal ileum.
The gut is so deeply interconnected. A problem in the stomach ruins absorption at the very end of the small intestine 20 feet away. Which actually brings us to a massive question. With all this food, all this fluid, all this acid, and all this bile sloshing around, the gut is constantly exposed to the outside world. It is a massive, warm, nutrient-rich, wet environment.
A perfect place for bacteria.
Exactly. How does it protect itself? How does it tell the difference between a harmless piece of bread, a beneficial probiotic bacteria, and a deadly salmonella pathogen?
To manage that threat level, the gut requires a massive, sophisticated military presence. The gastrointestinal tract actually contains the largest mass of lymphoid tissue in the entire human body.
We are talking about gut-associated lymphoid tissue. Its primary stated function is the immune surveillance of luminal antigens.
Yes, and the most prominent identifiable structures within the gut are called Peyer's patches. These are large, concentrated lymphoid follicles primarily located in the mucosal layer of the ileum, the final segment of the small intestine. You can think of them as embedded military bases.
And this is where we have to clarify a major confusing point in the biological terminology that the sources highlight. Earlier, we talked about M cells in the duodenum releasing the fasting hormone motilin. But overlying these Peyer's patches in the lower gut is a completely different cell. That, incredibly frustratingly, is also called an M cell.
It is a notorious point of confusion in physiology classes. The upper gut hormonal M cell makes motilin. The lower gut immunological M cell stands for Microfold cell. It does not produce any hormones whatsoever. Its job is purely immunological surveillance.
How does a microfold cell survey the gut?
Unlike standard epithelial cells that are covered in that thick, protective brush border of microvilli we discussed, these immunological M cells have a unique structure. They lack the microvilli,
They're bald.
basically. Instead, they have a deeply invaginated pocket on their basolateral side, the side facing down into the body tissue. This pocket is literally filled with active immune cells, T cells, B cells, and macrophages.
Okay, what do they do?
The M cell sits on the surface and continuously samples antigens, bacteria, and viruses passing by in the gut lumen. It engulfs them, transports them intact across its cell body, and essentially hands them over to the immune cells waiting in the pocket below.
Okay, instead of the generic border patrol analogy, I picture this more like a specialized interrogation room. The M cell grabs a suspicious character off the street, pulls them down into the hidden basement room, hands them to the detectives, the lymphocytes, and says, "Hey, have you seen this guy before? Is he a threat to the body?"
That is a perfect visualization. And if the lymphocytes interrogate the antigen and determine it is indeed a threat, they initiate both a localized and a systemic immune response. One of the primary, most effective defenses they mount is the mass production of secretory IgA.
Let's talk about secretory IgA. This is an antibody, right?
Yes, immunoglobulin A. But it is highly specialized for the harsh mucosal environment. When activated, B cells in the tissue under the gut lining differentiate into plasma cells and start pumping out dimers of IgA. But to be useful, this IgA has to get out of the tissue and into the dangerous enzyme-filled environment of the gut lumen.
How does it do that without getting digested?
It accomplishes this by binding to a special receptor on the bottom of the epithelial cell called the polymeric immunoglobulin receptor, or PIGR.
So the gut cell literally swallows the receptor and the attached antibody, transports it all the way across the cell, and spits it out into the gut cavity.
Yes, a process called transcytosis. And during that transport, a crucial piece of the receptor actually breaks off and remains permanently attached to the IgA. This is the secretory component. It acts as chemical armor, protecting the antibody from being immediately chopped up and degraded by all those powerful pancreatic proteases we discussed earlier.
That is so smart.
Once safely in the lumen, secretory IgA acts as a silent assassin. It binds to pathogens, neutralizing them and preventing them from physically attaching to the gut wall. But crucially, it does this without triggering a massive, tissue-damaging inflammatory cascade.
It just quietly takes the threat off the board.
Right.
But this brings us to what the sources suggest is the ultimate philosophical marvel of the gut immune system: oral tolerance.
Oral tolerance is arguably the single most complex computational task of the human immune system. Think about the systemic immune system for a moment, the white blood cells circulating in your bloodstream. Their default hardwired state is search and destroy.
Right.
If a foreign protein from a cow or a plant or a fungus gets injected directly into your blood, your immune system launches a massive, violent attack.
But wait, we voluntarily put pounds of foreign plant and animal proteins from cows and plants into our gut every single day.
Precisely the paradox. If the GALT in your intestines reacted to food proteins the exact same way the systemic immune system reacts to bloodborne invaders, you would have a nonstop fatal allergic reaction to every meal you ate.
That would be bad.
Very bad. Therefore, the GALT has an incredibly difficult job. It has to actively learn to suppress its own baseline responses. It maintains a state of systemic unresponsiveness to orally administered antigens. It has to constantly identify incoming, totally foreign proteins and confidently tell the immune system, stand down, this is just wheat, stand down, this is just chicken breast.
And while it is standing down and ignoring the food, it must simultaneously stay hypervigilant and ready to launch a nuclear strike on a norovirus or a salmonella bacteria. It is walking a razor-thin tightrope. And it gets even more incredibly complicated when you realize the gut isn't just an empty tube processing food. We have a massive population of permanent residents living down there.
The gut microbiota. While the stomach and the upper small intestine are largely kept sterile by the lethal gastric acid, and the constant sweeping of the MMC housekeeper, the large intestine or colon, is an entirely different ecosystem. It literally teems with bacteria, predominantly strict anaerobes, meaning they are organisms that survive only in environments completely devoid of oxygen.
The numbers listed in the sources are hard to fathom. An adult human colon contains roughly 1 kilogram of bacteria, comprising over 500 distinct species. That's over 2 pounds of alien life forms living inside you right now.
And they are not just passive hitchhikers or freeloaders. They are essential biological symbionts. We provide them with a warm home and food in the form of dietary fiber like complex cellulose from vegetables, which our human digestive enzymes are completely incapable of breaking down.
Right.
The bacteria ferment this fiber to survive, and in return for room and board, they provide us with profound life-sustaining benefits.
The sources note they produce essential vitamins for us, like vitamin K, which is critical because our dietary intake alone is often insufficient for our blood clotting needs. They also synthesize vitamin B12, thiamine, and riboflavin. But the most interesting byproduct of fermentation, to me, are the short-chain fatty acids.
Particularly one called butyrate. Butyrate is clinically fascinating. It serves as the primary preferred energy source for the colonocytes, the actual human cells lining the colon.
So our cells eat their waste.
Basically, they eat the bacteria's waste to survive. But beyond just energy, butyrate has powerful systemic anti-inflammatory effects. It actively supports the differentiation and function of regulatory T cells, or TREGs, within the gut mucosa.
TREGs are the peacekeepers of the immune system, right? They are the officers enforcing that oral tolerance and preventing the immune system from overreacting to the food.
Yes. So when you put it all together, the commensal bacteria are quite literally actively shaping, calibrating, and soothing your human mucosal immunity. They are manufacturing the chemical signals that tell your immune system to remain calm, tolerant, and balanced.
And what happens if you disrupt that?
When this delicate ecology is disrupted, say by a heavy, prolonged course of broad-spectrum antibiotics or a chronically poor diet completely devoid of fermentable fiber, you starve the bacteria. You lose the butyrate, you lose the TREGs, and that vital immune tolerance can break down.
Which perfectly sets up the final piece of the puzzle today: clinical pathology. We've spent the last hour building up this miraculous 30-foot, 250-square-meter automated processing plant. We've seen how it relies on exact pH balances to the decimal, precise hormonal timing from the fed and fasting teams, massive active enzyme cascades, highly specific molecular transport proteins, and an immune system walking a daily tightrope. What happens when specific parts of this intricate machinery break down?
The clinical consequences are swift, profound, and often devastating. And if you look at gastroenterology, the effects usually trace back to one of three categories of failure: enzyme defects, immune defects, or anatomical and mechanical defects. Let's look at one of the most common enzyme defects first: lactose intolerance.
This one is incredibly widespread. The source material outlines exactly what goes wrong here, and it connects right back to the architecture we discussed at the start. It's a failure at the brush border.
It is a localized, specific failure. The patient simply lacks sufficient levels of the enzyme lactase, which is supposed to be embedded on their microvilli. So when they consume lactose, which is the primary disaccharide sugar found in dairy milk, there is no detonator to break it apart. It cannot be cleaved into absorbable glucose and galactose.
So the complex lactose molecule just continues traveling down the tube intact. Why does a single sugar molecule cause such severe, painful symptoms?
For two highly disruptive reasons. First, osmosis. Lactose is a large osmotically active molecule. As it travels undigested through the small and large intestine, it drastically alters the osmotic gradient. It physically draws massive amounts of water out of the body's bloodstream and into the gut lumen, causing severe watery osmotic diarrhea and rapid fluid loss.
Oh, wow.
Second, fermentation. When that massive load of undigested lactose finally hits the 1 kilogram of bacteria living in the colon, the bacteria throw a feast. They are thrilled. They rapidly metabolize the lactose, and the byproduct of that massive fermentation is the production of huge quantities of gases, hydrogen, carbon dioxide, and methane.
So you get massive amounts of water rushing into the gut, causing diarrhea, while bacteria are simultaneously pumping the gut full of methane gas, causing severe distension, bloating, and painful cramps. It is a perfect storm from one missing enzyme. But as common as it is, it's just a localized issue. Let's look at what happens when there's a breakdown in that delicate immune tolerance we discussed. Celiac disease.
Celiac disease is a severe, systemic, immune-mediated functional defect. In a patient with celiac, the oral tolerance to one specific food protein, gluten, which is found in wheat, barley, and rye, has catastrophically failed. But it is crucial to understand that it's not a simple food allergy like a peanut allergy. It is an autoimmune reaction where the body attacks itself.
How did the mechanism actually work? The sources mentioned a specific enzyme called tissue transglutaminase, or TTG.
Yes. When gluten is partially digested by the stomach and pancreas, it leaves behind a tough, resistant peptide fragment called gliadin. In celiac patients, the enzyme TTG, which sits in the gut wall, chemically modifies this gliadin. This modified gliadin is then picked up and presented by specific HLA molecules DQ2 or DQ8 to CD4 positive helper T cells in the guild.
The immune system's commanding officers in the interrogation room.
Exactly. And because the tolerance is broken, the T cells identify this modified gluten as a highly dangerous, lethal pathogen. They launch a massive, localized inflammatory assault on the intestinal lining to destroy it. Over time, this chronic, violent inflammation leads to something devastating: villus atrophy.
Villus atrophy. So those millions of tiny vascularized finger-like projections we talked about earlier that multiply the surface area, the immune system literally burns them down to the ground.
It destroys the tennis court. The mucosa becomes completely flat and blunted. And because you have lost that massive absorptive surface area, you don't just lose the ability to absorb gluten, you develop global malabsorption.
Meaning everything is affected.
It affects everything. The patient loses the physical ability to absorb iron, folate, vitamin B12, fats, and the fat-soluble vitamins. They develop profound nutritional deficiencies, osteoporosis from lack of calcium, severe weight loss, and chronic diarrhea.
And the only treatment currently is complete removal of the trigger. A strict, lifelong gluten-free diet to stop the inflammation and allow the villi to slowly rebuild themselves.
Correct.
Okay, let's look at a different failure point. What if the tennis court is perfectly healthy, the immune system is tolerant, but the chemical demolition Cree simply never shows up to work? That brings us to EPI exocrine pancreatic insufficiency.
This occurs when the pancreas fails to deliver those vital digestive zymogens and lipase to the duodenum. The most common causes in adults are chronic pancreatitis, which is often, though not always, linked to heavy alcohol use that causes progressive scarring, fibrosis, and calcification of the organ over years. And in children, the primary cause is cystic fibrosis.
Wait, I'm stuck on that. How does cystic fibrosis cause pancreatic insufficiency? I always associate CF entirely with the lungs and breathing problems.
It is a systemic disease. The genetic defect in cystic fibrosis affects the CFTR chloride channels, which regulate salt and fluid secretion across all epithelial membranes. In the pancreas, this defective fluid transport leads to incredibly thick, dehydrated, viscous mucus secretions that physically plug up and block the pancreatic ducts. The pancreatic acinar cells are still dutifully producing all the zymogens, but they get physically trapped behind the mucus plugs and cannot flow into the intestine.
I use an analogy for EPI. It's like having thousands of dollars of high-quality raw materials delivered straight to the factory floor, but all the workers and machines have vanished. The body starves to death, not because there isn't food available, but because it physically cannot process it. The food just passes right through untouched.
That is exactly what happens clinically. And the absolute hallmark symptom of EPI is steatorrhea. Because the pancreas isn't delivering lipase, and the fats can't be broken down, the body cannot absorb fat. The undigested fat passes straight into the stool, resulting in pale, bulky, floating, and incredibly foul-smelling diarrhea. Furthermore, the patient rapidly becomes severely deficient in those fat-soluble vitamins A, D, E, and K, because without fat absorption, the vitamins are flushed away as well.
Which perfectly explains the classic exam question from the sources about the 45-year-old male with a history of heavy alcohol use presenting with floating, foul-smelling steatorrhea. The treatment is PERT pancreatic enzyme replacement therapy, where they literally have to swallow capsules of active enzymes with every meal to do the chemical work the pancreas can't.
Exactly right.
Let's move to anatomical defects. What happens when surgeons have to physically remove or bypass major sections of this 30-foot tube?
The physiological consequences depend entirely on what specific segment is removed, because, as we've seen, different segments have highly specialized, non-overlapping functions. Take the stomach, for example. If a patient undergoes a total gastrectomy for stomach cancer, or a Bill Roth II procedure where the stomach is surgically bypassed, they lose the function of their parietal cells.
No parietal cells means no stomach acid, but more importantly, no intrinsic factor. And as we discussed with absorption, no intrinsic factor means you cannot absorb vitamin B12 in the ileum.
Exactly. Even though the ileum is perfectly healthy and untouched by the surgery, the patient will inevitably develop pernicious anemia and severe irreversible neurological symptoms if untreated, simply because the stomach isn't providing the chemical key required at the end of the line.
What if you remove the end of the line? So you have to resect the terminal ileum because of severe Crohn's disease.
If you resect the terminal ileum, you physically sever the enterohepatic circulation. Remember, 94% of your bile salts are actively reabsorbed there to be recycled to the liver. Without the ileum, those bile salts spill over into the colon.
And what happens when a massive load of biological dish soap hits the colon bacteria?
Bile salts are highly irritating and toxic to the colonic mucosa. They stimulate massive water and electrolyte secretion, causing severe secretory diarrhea. Furthermore, because the liver cannot synthesize new bile fast enough from scratch to replace the massive daily loss, the patient's overall bile pool depletes. This secondary bile deficiency leads to fat malabsorption and, again, steatorrhea.
It is a brutal domino effect. And in extreme cases, like short bowel syndrome, where massive lengths of the intestine are removed due to ischemia or trauma, the transit time becomes so fast and the surface area is so small that the patient suffers from severe, life-threatening malabsorption of fluids and macronutrients, often requiring intravenous feeding for life.
Which truly highlights how dependent our baseline systemic health is on the precise sequential operations of this entire aerodigestive tract. If you look at rare genetic conditions mentioned in the text, like congenital chloride diarrhea or Hartnup disease, we see that every single microscopic molecule, whether it's chloride or specific neutral amino acids, relies on highly targeted specific transport proteins. If just one single gene coding for one specific microscopic transporter is faulty, the entire systemic balance of the body shifts, leading to profound disease.
Before we wrap up, I want to pivot to a really specific, almost situational pathology that the sources dedicate a surprising amount of time to. It's not a disease, it's not a genetic defect, and it doesn't require surgery. It's exercise-induced gastrointestinal problems.
Yes, modern sports nutrition highlights this as a major performance-limiting issue. The clinical sources note that an estimated 30 to 50% of distance runners experience severe intestinal problems during races. There's a famous quote from marathon legend Bill Rogers that captures it perfectly. More marathons are won or lost in the porta toilets than at the dinner table.
It's a massive issue for athletes. Symptoms range from nausea and heartburn to severe abdominal cramping, the urgent need to defecate, and even bloody diarrhea. Why does intense endurance exercise, which is supposedly healthy, wreck the gut so completely?
The clinical causes break down into three main categories: physiological, mechanical, and nutritional. Physiologically, it is fundamentally an issue of blood flow and survival priority. When you are engaged in intense, prolonged exercise, the sympathetic nervous system kicks in. It preferentially redirects blood flow away from the splanchnic circulation, which supplies the gut, and aggressively shunts that blood to the working skeletal muscles and to the skin for thermal cooling.
The sources say blood flow to the gut can be reduced by up to 80%. That is starvation.
That is massive, acute ischemia. The gut tissue is literally starved of oxygen and vital nutrients for hours while you are running. This ischemic environment severely compromises every function we discussed today. It halts motility, it stops absorption, and it weakens the tight junctions between the cells. It causes severe cramping, and in extreme cases, it can cause ischemic colitis, which is actual physical injury and cell death in the large intestine due to inadequate blood supply.
And on top of that ischemia, you have the mechanical causes. This is especially true in running compared to cycling.
Right, running involves repetitive, high-impact vertical jostling. Your entire GI tract is constantly bouncing up and down, slamming against the abdominal wall. This continuous mechanical trauma, combined with the fragile, ischemic, weakened state of the intestinal lining from the lack of blood, physically shears and damages the mucosa. This combination is believed to be the primary cause of gastrointestinal bleeding in runners, leading to occult blood in the stool or even frank bloody diarrhea post-race.
And then, as if the lack of blood and the physical bouncing weren't bad enough, athletes often inadvertently exacerbate the problem with nutritional causes.
Yes. Athletes consume highly concentrated energy drinks and thick gels to maintain caloric performance. But as we discussed earlier with gastric emptying, high osmolarity and high energy density severely slow the stomach down. If a runner consumes a hyperconcentrated carbohydrate solution, say an osmolarity well over 500 milliosmoles per liter, it doesn't absorb. It just sits heavily in the stomach, causing nausea.
Or worse, when it finally does squeeze into the ischemic intestine, that high osmolarity draws water out of the already dehydrated blood plasma and dumps it into the gut lumen.
Which causes explosive osmotic diarrhea, severely exacerbating systemic dehydration and instantly ruining the race. Furthermore, the type of carbohydrate matters. Fructose, for example, is absorbed much more slowly via that GLUT5 transporter than glucose. If an athlete relies exclusively on a heavy fructose gel, it overwhelms the transport capacity, remains in the lumen, ferments via the bacteria, and causes severe cramping and loose stool.
This is why if you've ever tried to chug a heavily concentrated sports drink during a hard run, it feels like it's just sloshing around in your stomach forever. The physiological recommendations are pretty strict. To prevent these issues, athletes need to avoid high-fiber foods for a day or two before the race. Fiber isn't digestible. It just adds physical bulk and bacterial gas, which you absolutely do not want when your gut is ischemic and bouncing. They also heavily recommend avoiding aspirin and NSAIDs like ibuprofen before a race, because those specific drugs have been clinically shown to increase intestinal permeability, making the mucosal damage and the bleeding significantly worse.
It is a perfect observable example of how stress, in this case, the extreme physical stress of running a marathon, forces the gastrointestinal system to the absolute breaking point. It demonstrates just how much energy, blood flow, and precise physiological coordination is required to comfortably digest a simple meal under normal circumstances.
Wow. Okay, we have covered a massive amount of ground today. We started by completely unfolding the small intestine, revealing a 250 square meter tennis court of absorptive architecture, heavily vascularized, perfectly folded, and actively churning.
We examine the brilliance of the enteric nervous system, the second brain operating autonomously as a smart home network with hundreds of millions of neurons to perfectly coordinate peristalsis and segmentation.
We watched the hormonal shift managers orchestrate the gut's behavior. We saw Modlin running the 90-minute housekeeping sweeps during the fast, and then the simultaneous rapid-fire signaling of CCK, secretin, and GIP the moment food arrived, perfectly managing the fat, acid, and sugar.
We delved into the chemical precision required for demolition, the extreme lethal acidity of the stomach, the ingenious evolutionary use of inactive zymogens to prevent autodigestion, and the vital role of biosalts and the highly efficient enterohepatic circulation.
We looked at the immune interrogation rooms, the GALT, the Peyer's patches, the unique sampling of the M cells, and the incredible complex ecosystem of the microbiome, actively teaching our immune system oral tolerance. And finally, we saw exactly what happens when those systems fail, from lactase deficiencies and autoimmune villus atrophy to pancreatic blockages and exercise-induced ischemia.
It truly changes how you perceive your own physiology. Your overall health is deeply, inextricably tethered to the flawless, automated operation of this 30-foot digestive tube.
It really is. And before we go, I want to leave you with a final specific thought derived from our sources today. It's something to really chew on, especially if you deal with everyday stomach issues. We learned that the extreme 0.8 pH acid of the stomach is the ultimate gatekeeper for the entire system. The sources explicitly state that patients with achlorhydria, a condition where they physically produce no stomach acid, can have up to 100 million microorganisms per milliliter living in their stomach instead of the normally sterile environment.
The acid is a vital, non-negotiable barrier.
Exactly. Now, consider everything we discussed today about the microbiome in the large intestine. Those specific bacteria actively train our mucosal immunity, they produce butyrate, and they maintain our regulatory T cells. They shape our systemic immune responses. So here is the question. Given that stomach acid is the critical filter determining exactly which bacteria survive the journey down into the gut to colonize our system, how much are we inadvertently rewiring our own systemic immunity and chemically altering the deep ecology of our second brain when we constantly suppress our stomach acid with daily over-the-counter antacids?
It is a profound clinical question. If you artificially alter the gatekeeper, you inevitably alter the entire downstream ecosystem.
Think about that the next time you picture that tennis court inside you. Until next time.